Hi everybody, it's your nerdy TA here. I am going to try to record again um, the video for going over histology of the digestive system with you, um, and hopefully I'll be able to keep it as short as I can. But I wanted to review with you um, how what sort of things we want to be looking at when it comes to histology. And for those of you who are going to be following along with your lab manuals, it's going to be over pages 58, 59, 60, 61, and a little bit of 62 in your lab manuals. Okay, so let's get started. So first I want to lay out a basic um, template for you to look at when we're walking through this. And the basic template that it's pretty much going to follow through the alimentary canal, which remember that's the route that our food is going to take as it goes through our digestive system, right? So, in histology photomicrographs, we want to start by identifying where the lumen is. And once you found the lumen, from the lumen to the externalmost layer, we're going to see the mucosa, the submucosa, the muscularis externa, and depending on whether it's intraperitoneal or retroperitoneal, we're going to call it either serosa or adventitia, right? So hopefully that's a little bit of review from before we left for break. Now you may need to know that the mucosa has three different layers. So if a question is pointing to the epithelial layer, um, that's not going to be the mucosal layer. The mucosal layer is going to include your epithelium, but it's also going to include your lamina propria as well as your muscularis mucosa, which is a very thin layer of smooth muscle. Um, so uh, I'll show you that on some of these slides. And then the submucosa is certainly going to be staining just a little bit more lighter and whiter than some of our other layers. Here we're going to see some areolar uh, connective tissue and maybe some fibers, and you might see blood vessels or nerves even. And that's true for both the lamina propria and the submucosa. And what you'll find is that the lamina propria and the submucosa, there's actually quite a few similarities um, between them. But this is really where you're going to find your lymph vessels and your malt tissue and all that. And I really want you to keep your eye out for the submucosal layer in some of these slides because um, the submucosa is a great way to identify the differences between some of these um, parts. So then the muscularis externa, I'll point that out, but most of the time you're going to have an inner circular layer of smooth muscle and then an outer longitudinal layer of smooth muscle. And if it's intraperitoneal, we're going to call it serosa, but if it's retroperitoneal, then we're going to call it adventitia. Okay. So one thing I want you to keep in mind is what kinds of muscle tissue we're going to be using to perform certain types of motion, right? So skeletal muscle was responsible for voluntary movement, and smooth muscle was the muscle that's responsible for involuntary movement. So most of the digestive system is going to be filled with smooth muscle in the muscularis externa, and um, there's only a few places where you'll see some variation in that. Um, so one thing that might be helpful for lecture but not lab, actually I didn't see it anywhere in your lab manual, but one thing you might want to know is that peristalsis is that pushing of your uh, contents, your digestive contents through the alimentary canal. And that's going to involve both the circular and the longitudinal smooth muscles contracting. However, segmentation is going to involve your circular smooth muscle only. Okay, let's see. All right, so let's get started. So I would like to start with the superior most um, part of your alimentary canal as far as the histology slides that are in your lab manual. And the first one is the salivary glands, which actually starts on page 61 of your lab manual. And you need to know, so you wouldn't be expected to identify the differences among the different glands. So you wouldn't need to know, is this the parotid gland versus is this the sublingual gland versus is this the submandibular gland? You would need to identify those structures as far as gross anatomy goes, but as far as histology slide goes, all you would need to be able to do is just identify, is this a histology picture of a salivary gland? And there are three different parts that you need to be able to identify. That's serous cells, mucus cells, and duct cells. So next to serous cells, I would like you to write down watery. Okay, and these serous cells are going to be responsible for secreting enzymes, and they're only really secreting these digestive enzymes before and after food enters. So it's going to have a more watery secretion, whereas these mucus cells, I want you to write down next to it thick, viscous. And these guys are going to be secreting a viscous sort of mucin, right? So remember with goblet cells, how they produce mucus to help with lubrication. And we saw that a lot of times when we look at epithelial cells, goblet cells also stain a little lighter, right? So mucus cells are going to stain a little bit lighter, as you can see here, and the secretions that they produce will help keep the mucosa moist, and so they're pretty much released continually. And our salivary glands are composed of different amounts, different ratios of serous to mucus cells, so they differ slightly in that way. In some upper level classes, you may be expected to know that based on the histology slide, but for this one, you just need to know which ones are, which cells are which. And then finally, we have duct cells. And duct cells here are very easy to identify compared to serous and mucus, I think, because um, you can really clearly see a circular layer of simple cuboidal epithelial cells with a little hole in the middle. And these duct cells, remember, are going to, these are um, exocrine glands. So exocrine glands means, endocrine releases hormones into the blood, but exocrine glands are going to release secretions through a duct and out into a space or some sort of area. And so that's what these guys are going to do. The salivary glands are going to um, gather these secretions produced by these serous and mucus cells and route them through the ducts and out into the oral cavity. So anytime you see these circular, simple cuboidal cells, you can pretty much tell, okay, that's going to be like a door. Um, Now, I will say that it may be um, easy to get a close-up of a salivary gland histology slide mixed with possibly a histology slide of the pancreas. So two major differences I want you to make sure that you know is one, You're really not going to see a whole lot of this like this in the pancreas slide. So these are adipocytes, right? These are completely white lipocytes because remember, lipids don't take stain very well, right? It's just very greasy, it just kind of slides off. So um, you can see these adipocytes, but also in the pancreas slides, make sure you're looking for those islet cells, right? That cluster of islet cells. So if you don't see that and you see these adipocytes, chances are you're looking at a salivary gland. Okay, so here's another nice close-up. As you can see here, you can clearly see in the sear cells like a very dark nucleus, and the sear cells are also staining a little bit more darker, whereas the mucus cells stain a little bit whiter because of that thick um, lubricating mucus that they produce. And here is our circular ring of duct cells, right? Very clear, simple cuboidal around a circle. Okay, next we have our esophagus, which is on page 58 of your lab manual. And I want you to remember to take note of where the esophagus is in relation to some of the other structures around it. So don't forget the esophagus is posterior to your trachea, right? Because we just covered the respiratory system before this, and both of those systems are going to be on your next lab practical. The mucosa of the esophagus is your dead giveaway. Look at that. That is so thick. It's so thick. It's got a huge, thick layer of stratified squamous epithelium. Now, try to think back to where you might have seen stratified squamous before. And if some of you were thinking skin, you'd be absolutely right. Brilliant, brilliant people. So remember, skin had stratified squamous keratinized epithelium. This is non-keratinized, but it sort of serves a very similar purpose, right? We wanted very thick layers of squamous cells for protection, and that's exactly what you want to do with the esophagus. When you take that giant bag of Doritos that you just pretty much inhaled and didn't thoroughly chew up, you don't want it to be scratching and really being abrasive to your tissues as it makes its way down. So you want a very thick layer. So as soon as you see this, I want you to think esophagus. Okay, and then sort of scattered throughout your mucosa and submucosa, but really you're going to see more of these in your um, submucosa more likely, are your esophageal glands. And your esophageal glands are going to be responsible for secreting a lubricating mucus. Because remember, the esophagus, its major goal isn't to aid in digestion in terms of secreting major enzymes or absorbing major things. It's just moving things along to the next station. Um, so its mucus is simply just going to be responsible for lubricating. Now, in your lab manual, it says on page 58 at the bottom, what type of muscle forms the muscularis externa of the esophagus? So I'm going to take the time to explain to you now, and this is something you definitely want to write down if you haven't already read this in your textbook. There's one super unique thing about the esophagus that is different from all the other parts that you need to know for this unit. And that is that the muscularis externa of the esophagus is actually split into thirds. Okay? So we had said most of the alimentary canal, the muscularis externa is going to be composed of smooth muscle in the muscularis externa. And that makes sense. Digestion is involuntary. But think about what happens when you swallow. When you swallow, the first part of that is voluntary, isn't it? So it's actually split into thirds. The superior third of your esophagus is actually going to have skeletal muscle in your muscularis externa. The inferior third is going to be composed of smooth muscle, very much like the rest of your alimentary canal. And the middle third of your muscularis externa is actually going to have a mix of both skeletal and smooth muscle. So that's super unique, but it's also really cool if you think about it. It's like, wow, that it can, even within this one organ, it can differentiate the types of smooth muscle it has for best fitting that function. And don't forget, because the esophagus is retroperitoneal, we're going to call it the outermost covering, the adventitia. Okay, so this is a beautiful transverse cut of the esophagus, and I love this picture because you can see the different, just the stains that they use are so beautiful here. You can see that this is the mucosa. Remember, mucosa is not only the epithelial layer, right? So there's your epithelial, boom, super thick stratified squamous epithelium. You've got your lamina propria, and you've got your muscularis mucosa. And take a look at these this submucosal layer. See how it stains a little bit wider and lighter, thanks to all those things we talked about before. And then I love this. This muscularis layer shows it, this picture does a great job of showing you the different orientation in the muscle fibers. You've got an inner circular layer, and here you've got your outer longitudinal layer and your adventitia. So that's a beautiful dead giveaway for esophagus. So if you were shown a histology slide like this, you could be asked potentially which third or which area of the esophagus might you be looking at, superior, middle, or inferior? And the way that you can tell is that there's a presence of both smooth muscle and skeletal muscle, which should be your dead giveaway for the middle region of the esophagus. And don't forget, this is, I mean, you can definitely tell this is your mucosa, but the difference between your submucosa and your muscularis externa, remember, you're going to have a lot less interstitial space. And so you can think about it like this is like your Kroger bacon, and this is like your Whole Foods bacon for the super, super healthy people. Okay, so this is the lowest region of the esophagus. This is where the esophagus meets the fundic region of your stomach, the very beginning of your stomach. And don't forget that the sphincter you need to know there is your, some books will call it your lower esophageal sphincter, and some books will call it the gastroesophageal sphincter. Either one is fine. Go with the one that's in your tag list, which I think is gastroesophageal sphincter. Okay. And I can guarantee you really should look at multiple different sources of slides for this junction. This junction is super important. This is really like a great way of seeing how form relates to function and how that changes through your body. Um, and the big thing is that where it turns from the esophagus to the stomach, that's going to be your junction. So here you can see that there's a super thick stratified squamous layer, and here you can see that the epithelial layer goes from super thick to very, very thin. And so in the stomach, we're going to find simple columnar epithelium because we're not worried so much about protecting from abrasion. We are more concerned about secreting certain things in our gastric pits and making sure that we have plenty of goblet cells. So you're going to see lots of goblet cells along this columnar epithelium. And so it's a very, very clear divide between the two. Here is an even better stain where you can see the color difference more clearly. Here is a very thick stratified squamous epithelial layer. And here, look at those lovely gastric pits. I mean, come on, that's you can just see how there's folds in this mucosa where you're going to see some of these gland secreting cells um, and all these goblet cells. Okay, so make sure you look at a couple different ones like that and quiz yourself. Here's the esophageal, gastroesophageal sphincter gastroesophageal junction again. And like I said, once it's really, really zoomed out, you should still be able to tell, boom, very thick, stratified squamous. And then all of a sudden, when we get to here, boom, very thin looking simple columnar epithelium. Okay. One major thing that you should know that the stomach is sort of unique in compared to the rest of the canal is this is pretty much the only place you're going to find 3 different orientations of your muscularis layer, of the structures that you need to know for this exam. And like I said before, most of the time we're going to find a circular layer, an inner circular layer and an outer longitudinal layer, right? Well, guess what? This is how you can tell where if you're in the stomach. You're going to have an innermost oblique layer. So why do we have an oblique layer? Remember, think back to A&P1, you learned that muscles and how the different arrangement of the muscles were sort of what told you the direction of movement, because how it contracts can tell you how it's going to be moving, right? So that's exactly what's happening here. The oblique layer of your muscularis is responsible for helping you with the mechanical digestion that the stomach's going to do, called churning. And it's really going to just create a big sort of mixing, tumbling motion that's going to thoroughly mix your bolus with the gastric juices to make that chyme. Okay, so another thing is that you're going to need to know the 4 different cells you can find in your gastric pit. And on page 59 of your lab manual, and that includes chief cells, GNES cells, mucous neck cells, and parietal cells. Chief cells are going to make pepsinogen, and I say ogen because it secretes it as an inactive form, and it'll get activated into pepsin later. And then it's going to secrete gastric lipase, which does a little bit of your lipid digestion, which you'll learn about in lecture. Your GNES cells, another name for them might be enteroendocrine cells. Let's see if I have a... No? Okay. Your enteroendocrine cells, and those are going to be more of your hormone producing cells. You might hear them used interchangeably as G cells, but actually G cells are technically a specific type of enteroendocrine cells. And what they're going to do is secrete gastrin. And a lot of times you're going to find those enteroendocrine cells closer to the bottom of the pits because you want to give them easy access to the blood vessels down below because they want to secrete that gastrin into your blood. Then the mucous neck cells, you actually have surface mucus cells and mucous neck cells. They both produce mucus. And here's the difference, and this is super cool. It's one of my favorite things about the stomach. You have surface mucus cells out here, and then you have mucous neck cells in here. And this is like the neck area of your gastric pit. The surface mucus cells secrete a more alkaline mucus. And if you think about it, that makes sense because what's one major thing we're going to be secreting here in our parietal cells? Our parietal cells are going to be secreting intrinsic factor, which you need to help you absorb vitamin B12 in your small intestines. But you're also going to be secreting hydrochloric acid. Now, in lecture, side note, you are going to learn about how it's not really secreting hydrochloric acid like preformed. It's actually just pumping protons into the lumen, and it's going to bind with chloride ions to make hydrochloric acid. But regardless, those parietal cells are helping you make that acidic environment, right? Those acidic juices. Stomach juice has a pH of 1 or 2. That's incredibly acidic, okay? And that can be really, really damaging to your cells. So that's why you want an alkaline mucus layer here to make sure that's nice and protected. It's kind of like if any of you have ever watched those alien movies, when they start like spewing acid in your face, it kind of like melts your face, right? Yeah, you don't want that to happen to your inner tissue. So you really want to coat that with a nice alkaline mucus that'll keep it neutral and protected. But then think about this. If you're making mucus down, making hydrochloric acid down here, and you want it out here, well, you don't want to make alkaline mucus here, because what are you going to do through this opening before you get out here? You might neutralize that acid. You don't want that to happen. So the mucus neck cells will, yes, they do secrete mucin, but they secrete a more acidic mucin, whereas your surface mucus cells will secrete an alkaline mucin. So that's super cool. I mean, even on a microscopic level, the cell differences can show you the differences in function. So um, this is a dead giveaway for our looking at uh, parts of the stomach. So you're going to need to know that, this, okay, find your lumen, boom. Then the black bracket is your mucosa. Then your blue bracket is your submucosa. Then your green bracket is your muscularis externa. Then your green arrow, based on once you identify it's the stomach, we can say, oh, aha, that's going to be our serosa. Okay, and look for these clear oblique, the innermost layer will be oblique muscle. Okay, so then we get to our small intestine, and big difference in our small intestine. So um, you're going to need to know the difference between how can you tell when something is a circular fold versus when something is a villus versus when something is a microvillus. So a circular fold is going to be pretty visible on a microscopic picture. And if I can go here, this whole thing that my arrow is pointing at, that is a circular fold. It's called a plicae circularis. And this is the largest type of fold. It's quite visible. It's like a big fold of your mucosa as well as your submucosa. And if you look very, very closely, there are all these villi on the surface right here. So each villus is one of these hairy looking things. Now, you cannot see a microvillus on a magnification like this. Okay, you can only see it when you go super, super, super up close. Okay, so if you're given a slide like this, you're not going to expect to see a microvillus. So, if they're pointing to one of these and asking you what it is, do not put microvillus. Okay, you can only see a microvillus from something as detailed as something like a transmission electron microscopy. That's how close you have to get to be able to see it, because what it is it's tiny folds of your cytoplasm, of your uh, not not cytoplasm, your plasma membrane. I just had a brain fart there. Your plasma membrane of the enterocytes. So you're gonna have hundreds, sometimes thousands of these things on each enterocyte. So again, you really can't see it till you get to an electron microscope. So here you can see a lot of goblet cells. Each one of these is a villus, so multiple of these are villi. Um, this is also where you're going to start to see some of your lymph vessels in your submucosa, right? And this structure is called a lacteal. And you're going to learn about in lecture about how the lacteal is, we said a lymph system is responsible for a lot of our immune function, but these lacteals also help in lipid absorption, because those things are very, very difficult to get into our circulatory system. So you'll learn about all of that. Okay, so this is from your histology guide.org. And actually, let me see if I can turn to that in a second. But in this zoomed out view, these are three parts of our small intestine. We've got our duodenum, we've got our jejunum, and we've got our ileum. So how can you tell? Well, I've been told that you're not going to be given a histology slide super zoomed in on just the villi because it's very difficult to tell from just the villi unless you've done this for many, many years, and it's very difficult to tell the difference between this villus versus this villus. However, you will find a greater frequency of plicae circularis in your duodenum. You will see some of it in your jejunum, but by the time you get to your ileum, you're really not going to see those circular folds. Also, in your duodenum, you can expect to see, look in your submucosa. They have to give you a picture of the submucosa if they're going to give you the duodenum. And in the submucosa, you're going to find these duodenal glands called Brunner's glands. And this is pretty much the only region of your small intestine where you're going to see these glands in the submucosa. And it's going to help secrete like a lubricating fluid again. And I find, in my opinion, these the villi of the duodenum seem a lot more regular to me versus the villi of the ileum tend to look a little bit more regular and a little stumpier because you don't see um, those circular folds like that. And if you don't see any glands, if you don't see, one thing I want you to look for in the ileum are these regions in the submucosa. What is that going to be? We talked about that in the lymph system unit. That's going to be our Peyer's patches with malt tissue, right? So if you don't see Peyer's patches and you don't see duodenal glands, chances are it's going to be your jejunum. Okay. All right. So um, let me see if we can go back out to our histologyguide.org and looking at our small intestine here. Okay, so there is our duodenum. And when we get right up in there, look at that. These are Brunner's glands, our duodenal glands in the submucosa. See how they stain a little bit lighter? It's because they're going to be secreting like that mucus-like substance, right? So that is something you are not going to see in something like your jejunum. So let's zoom in into the jejunum. See that? They are not stained lightly. You are not going to see duodenal Brunner's glands in your submucosa. Okay, so now we go to our ileum and we will zoom in. See how the villi are a little bit shorter, broader, and I think you can really see well on this particular section, boom, Peyer's patches, right? It's going to be filled with lymphoid tissue and uh, malt, malt tissue. Okay. All right. So back to this. Our large intestine is very different because you're not going to see villi anymore. You're not going to see villi. You're not going to see plicae circularis. However, what you are going to see is a much higher frequency of goblet cells. If you see goblet cells as far as the eye can see, you can be guaranteed you are in the large intestine. Are you going to be expected to know the different parts of the large intestine? Like, are you going to be given a histology slide and ask the difference between, say, the ascending colon versus the descending colon? Absolutely not. All you need to be able to identify is this the large intestine? But in the large intestine, you can tell this apart from the small intestine because you can look for big, large spaces in what you might think might be your villi. But if you don't see that, they're not your villi. So um, really what you're going to see is these simple columnar epithelial cells. You're going to see your very thin submucosa, and then your muscularis. Um, you also are going to see some intestinal glands that may extend into your muscularis mucosa. Um, and remember, you're going to have less and less water in your digestive contents as you go down the tract. So um, by the time you get to your large intestine, your small intestine has already done a majority of your absorbing of both nutrients as well as water, electrolytes, and all that jazz. So that means the more water you're taking out of it, the more uh, potential for pathogenic exposure your cells can have to these stomach contents, right? So it's super, super important to, number one, you have less water, to make sure that you're lubricating it so that you can move it nice and smoothly through your large intestine. Number two, make sure that you're protecting your cells from getting infected, because what else are we going to have in our large intestine? We're going to have tons of normal microbiota. What does that mean? That means we have bacteria in our large intestines, and they're super important. They're not there to harm us, they're there to help us. Um, and they are going to help make some of these vitamins that we need, and help us with some of that absorption. So, lots of goblet cells, but also you're going to see more lymphatic nodules and lymphatic cells in your lamina propria. And your smooth muscle, again, you are going to have an inner circular and an outer longitudinal layer, but in some of these slides, you might be able to see the outer longitudinal layer will be discontinuous in your large intestine. And in some of these, you might be able to see that the longitudinal smooth muscle fibers are actually going to be forming your Taenia coli. So here's a good side-by-side view of your esophagus, which you can tell um, by your stratified squamous. Now, one thing I think you can get mixed up on if you're not careful is the stomach versus large intestine. So don't go simply off the fact that you see flat tops, because that's not enough. Both of these have them in different parts. Like the fundic region is going to have a lot of flat tops. But I want you to see rather, okay, do you see an oblique layer of smooth muscle in your muscularis externa? You can tell these are going to be gastric pits, right? They're kind of congregated at the bottom of these pits here, and they're kind of stained a little bit darker. Whereas here, you have these nodules, which are probably lymphatic tissue, and you're not going to see that just sort of secluded and contained at the bottom of these pits like you would the stomach. And the small intestine is very easy because you see this villus. Um, this is a great side-by-side view of parts of the small intestine versus the large intestine. You can definitely tell the difference from the colon with the rest of them because there's just way, way, way more goblet cells. No villus, right? Almost looks like a fuzzy layer here. However, will you be expected to know the difference given slides like this at the bottom row between the duodenum and jejunum, or between the jejunum and the ileum? Absolutely not. You have to be given the submucosa so you know what to look for. You are more likely to be given something like this, like the top row for the duodenum versus like the ileum, say, um, or the duodenum versus the jejunum. And don't forget, and this even this picture of the jejunum is not all that great, but one dead giveaway that this is the duodenum is those duodenal glands, the plicae circularis, and then in the ileum, those Peyer's patches. Okay, so this is a great way to test yourself. Um, And I would go through this whenever you get a chance. So think about what these mean. Number 3 is difficult, and you wouldn't be given a slide like number 3, but I'll go ahead and just walk through this one with you now. This would just be your small intestine. Um, in my opinion, I think it could be the jejunum because I don't particularly see Brunner's glands, and I don't particularly, you can't really see the submucosa very well, but I don't particularly see Peyer's patches, so that's why I would say jejunum. But this is not the best picture, and you probably wouldn't be given something like this. It's a little bit more ambiguous where you can't clearly see the submucosa. And then the rest of them you should be able to get. So I would go ahead and try those whenever you get a chance. Some accessory glands you're going to need to know. We already talked about the pancreas back in the endocrine unit, but keep in mind that this is how you can differentiate the pancreas from the salivary glands, is these islet cells. Now, the islet cells before were what we focused on because that was the endocrine unit, because we're concerned about secreting hormones, right? So who's the star of the show now? Who's your MVP? These are your acinar cells, okay? So each of these is an acinar cell. This is an acinus, and these are acini. And these guys are going to be secreting that uh, that pancreatic juice portion that's made up of bicarbonate ions. So it's going to be like a basic alkaline type secretion. I'm sorry, that's what your duct cells are going to do. Your acinar cells are going to be secreting digestive enzymes. So those are the two stars of the show, the duct cells and the acinar cells. The duct cells are going to be sort of around these duct entrances, right? So the duct cells are also going to be simple cuboidal epithelial surrounding the duct entrance, whereas your acinar cells are certainly going to be they are also simple cuboidal, but they're modified simple cuboidal in these little clusters. So that's how you can tell. And the duct cells are the ones that will secrete the bicarbonate ions, the base alkaline part of the pancreatic juice, whereas your acinar cells are going to secrete your digestive enzymes. You're also going to need to know, be able to identify the histology of the liver. And what you need to know about the liver is you got to be able to identify a lobule. And the lobule, I would just look for this hexagon here. And that is a single liver lobule. It's the basic unit of the liver, and it's separated by a little septa, connective tissue capsule. These are filled with tons and tons of hepatocytes, and they're all the lobules are kind of stacked on each other. If you look back at those classroom models you worked on, the center of the lobule is going to contain a central vein. So it might look white here on some of these slides, but that's where your central vein is going to come through. Then you've got your corners of the lobule, and on each corner are these little clusters, and I like them because I like to think of like a bunch of liver, like Chinese gangsters. It's your portal triad. So you've got your hepatic arteriole, which is a branch of your hepatic artery. So thinking back to blood vessel unit, the hepatic artery is going to provide oxygenated blood to your liver. Then you're going to have your portal venule. So there's our red right here. And obviously they're not going to be red, blue, and green on a histology slide, right? But just for looking at this diagram for now, this is your hepatic arteriole, bringing oxygenated blood. Then you've got in the middle, you've got your portal venule, which will be a branch of your portal vein. And remember, your portal vein is going to take some of that the hepatic portal um, the hepatic portal vein is different from your hepatic vein, right? So the venule is actually going to be a branch of your hepatic portal vein. So that's the one that's before the liver that feeds oxygen poor, nutrient rich blood into the liver. And remember, hepatic veins without the portal, just the veins, are the ones that take away the deox blood, the used blood from the liver back to your inferior vena cava. Then you have this green vessel right here, and that's a little bile ductule. Um, and one major thing you need to know is that bile is going to flow the opposite direction of blood. So the blood is going to be flowing this way from the vessels towards the center here. And I want you to watch this amazing, amazing video of sort of the layout of the liver, but you're going to see that the bile flows towards the portal triads here. So it's super cool. So that's the video that I put, and you can go and watch that. But basically, as you walk through this video, I want you to be able to see that the blood is going to flow from your hepatic arteriole and your portal venule to these hepatic sinusoids, which are these large, leaky, fenestrated capillaries that are going to pass between the rows of your hepatocytes. And blood is going to move very slowly through the sinusoids because you want materials between the blood and the hepatocytes to be exchanged. And then you're going to go from your hepatic sinusoids to your central vein in the middle of that lobule. And then you're going to, all the central veins are going to merge and they're going to drain into your hepatic veins up here in the purple. And that's going to feed back into your inferior vena cava in the back here in blue. So the hepatic sinusoids are going to be involved in that blood movement, whereas bile flow through the liver lobule is the opposite direction. And so these hepatocytes are going to help secrete that bile. And keep in mind, remember, your liver is the one who's making the bile, not your gallbladder, right? Your gallbladder only helps you store some of it and concentrate it. It doesn't actually create it. So your hepatocytes of the liver are creating the bile, and the bile is going to travel through your bile caniculi. These are tiny ducts. A single duct is a caniculus, but it's going to go through the bile caniculi and empty into the bile duct, which is going to empty into some of the other ducts we talked about with the pancreas and whatnot in gross anatomy. So here's another picture to show you that if you're given a picture like this is how you can tell this is your portal triad. You have 1, 2, 3. The biggest one in the middle, hepatic portal vein. It's taking deoxygenated, nutrient-rich blood that it absorbed from your digestive organs. It's going to be massive. Then you've got your bile ductule, and you've got your hepatic branch of the hepatic artery. Okay, so the blood is going to all flow into the central vein here. So this whole thing is a beautiful hepatic globule. Okay, so this is just like a nice sort of zoom out to show you, to tie this back into the circulatory unit. Remember, small intestine, large intestine, spleen all feed into the hepatic portal system. We're in our liver. Our liver is going to filter that blood. It's going to process it. It's going to drain that blood into the inferior vena cava through the hepatic veins. Okay, and that's pretty much it. Okay, so I think this is a wonderful little, let's see if I can Go to the read mode. Okay, well, that's fine. Hmm. Okay. Oh, well, anywho. So this is a nice little dichotomous key to kind of show you like a sort of a standard way to look at how to differentiate what you're looking at. So do you see goblet cells? Yes or no? If it's Now you will see goblet cells on some of the stomach slides, so this isn't perfect, but you are going to see certainly much more of them. So this really should say, do you see lots of goblet cells? And if the answer is yes, that is going to be your small intestine. Um, and for some reason, my mouse is not really cooperating all that well with me, so I will just go ahead and take a snapshot of this and send it to you guys. But these are some of the things that you can walk through. Okay, do you see some mucosal glands? Do you see um, Brunner's glands? Do you see crypts? Things like that. Crypts are those folds. They kind of look like gastric pits a little bit, but those folds in the large intestine. Okay. And don't forget to practice on histologyguide.org, as well as your Mastering A&P website. If you go to Access Study Area, and then if you go to Lab Tools, and you go to Practice Anatomy Lab, PAL, and go to Practice Anatomy Lab 3.1, and you go to Histology. It's amazing. Please make sure you're practicing with this, okay? If you scroll down, you can go to Digestive System. And you can either show labels or not show labels. If you want to show labels, you can. I would consider that as sort of your first training wheels. But really, when you're ready to take the training wheels off, um, I would make sure you're really trying to practice recall. You can go to your quiz and you can try some of these um, as a multiple choice. Uh, and then you can, when you're really, really ready to take all the training wheels off, you can try the lab practical portion where you can actually type in your answers and check your spelling. So please, please, please make sure you're practicing with them with regularity. If you have any questions, feel free to shoot me an email, and I will see you next week.